Next, we have Meizhen Han from Beijing Normal University presenting Precise Individualized Infant Brain Atlas. Thank you.
Hello, everyone. Today, I would like to introduce our recent work on infant brain atlas construction. I have no disclosures. Let me introduce some background first. Cortical parcellation is a fundamental tool in brain area localization, network analysis, and cross-study comparisons. And it can also help to reduce data complexity and improve statistical sensitivity. So it's very important to construct brain parcellations. But while adult analysis are well established, they are not suitable for infants because of the differences such as brain size, shapes, connectivity, and function. Besides, infant brain represent rapid structural maturation in synaptogenesis, white matter fiber microstructure, myelination, and connectivity networks. Existing pediatric neuroscience have construct atlases focused primarily on neonate stage or functional modality, leaving a gap in structural connectivity-based parcellation. So in this study, our major goal is to fill this gap. There are some necessary challenges in construction of the infant brain atlas. The first one is dynamic and drastic brain development during the first 2 years after birth. And the second one is large intersubject variations in brain topography. So what's our solution? For the first challenge, to highlight the rapid development in the early infancy, we adapt an age-accelerate sampling design to construct densely sampled age-specific group atlas monthly to capture the developmental progress. And for the second challenge, we integrate individualized atlas technique into group atlas construction framework. Here is the dataset information. We include 2 independent datasets in this study. The first one is parcellation dataset, which consists of brain images from Baby Connectome project. After quality control, 464 scans from 170 infants were included. We stratified all scans into 16 detailed age subgroups with sliding window strategy. Here are the sample distributions across 16 age subgroups. And here are the scanning parameters of the DMRI data in dataset 1. We also perform some validation and analysis across different existing atlases. So we include independent dataset 2, which contains 31 typical developing infants data. To construct this infant atlases, we proposed fiber architecture-based atlas generation approach. There are 3 main stages in this framework. In stage 1, we first performed voxel-wise tractography, and then we calculate voxel-wise brain connectivity matrix under the spatial constraints from white matter bundles. In this matrix, in this matrix, each element represents the probability of the fiber tracks passing from specific voxels through each white matter bundle unit. And we also calculate similarity matrix. Then by applying spectral clustering on the similarity matrix, we can divide the DKLI into several subregions. But here, we cannot decide the exact number of subregions. So in stage 2, we evaluate the optimal clustering solution key separately in each age group for each DKLI. We produce a dual matrix strategy by jointly considering the reproducibility and clustering performance. Here we can see in this figure, the blue line represents the reproducibility, and the purple line represents the clustering performance. And we can find the patterns are quite consistent across different age groups. So we are able to achieve consistent number of subdivisions and scaling and the label scheme through infancy. Then in stage 3, we aggregate all the initial parcellation from all infant scans to generate model 0 antlers. And then we refine the parcellation through iterative group individual alignment within each age group until convergence. Here are the age-specific group-level probabilistic infant brain atlases. Here we can see the growth spatial distribution was consistent through development, with refinements at the region's edges. Then we perform cross-antlers comparisons to validate our proposed antlers. Specifically, we compared the homogeneity against other existing antlases using validation dataset. The homogeneity was defined as the average Pearson's correlations of white matter connectivity profiles between all pairs of voxels within a given parcel. Here we include 7 existing antlases, which comprise major categories of microscope brain antlases, such as classical anatomical parcellation, manual delineated parcellation, and functional parcellations, and multimodal parcellation. And we also include white matter connectivity-based atlas for adult version. These atlas are all included in the comparison. But as we know, many previous studies have showed that atlas homogeneity will strongly depend on parcellation granularity. So for fair comparison, we used a non-model approach, and we measured normalized homogeneity, which was defined as the ratio between the homogeneity of the parcellation and the random parcellation. Such normalization improves comparability across antlers with varying resolutions. Here are the results. We can find our proposed age-specific infant antlers have greater normalized homogeneity than all compared antlases. Then we also construct individualized brain antlers with spatial constraints from the newly proposed age-specific group antlers. First, we use ICC to estimate the test-retest robustness of the individualized antlers from repeated scans. We can see in this field map, the SECs are quite consistent across different age groups, and with a mean SEC is equal to 0.73. Then we track brain atlas growing using longitudinal data. Here we can see here, if we take the last scan of each longitudinal babies as reference, we can find a clear convergence toward reference antlers during the development. Then we also evaluate the capability of our individualized infant antlers to encode brain maturation. We use generalized additive mixed model to estimate the developmental effect of brain connectivity, homogeneity, and also subregion size ratio. And we find heterogeneous, nonlinear developmental trajectories across different brain subregions. Last but not least, we also perform sensitivity analysis. The split half validation demonstrates the reproducibility of group antlers can reach 95% with only around 30 samples. And individual antlers have better reproducibility, around 90% with spatial constraints from as few as 10 samples as previous. In summary, we proposed fiber and we generate the first densely sampled age-specific structural connectivity antler series for infancy. And it has been a validated tool for individualized mapping and can encode brain maturation during development. We are preparing for public release, and we hope it can serve as a common tool in pediatric neuroscience in a very near future. I'm grateful for Professor Yunhe and Teng Daizhao and all lab members, and we want to thank the entire BCP team and participants and their families for the invaluable contribution in data resources. Thank you.
We only have time for one quick question, either from the microphone or from the app. So from the app, there's a question regarding whether you're considering now extending your work to neonatal atlasing.
Yes. The parcellation data set in our study, the infant aged from 0 months to 26 months. So in this antlers, we have included neonate stage. Thank you.
Thank you for clarifying. Thank you again.